So now we transition to active processes. Now, active and passive processes are slightly different. With passive processes, we did not require energy. With active processes, we do require energy. And so active processes are organized into active transport and vesicular transport. With active transport, we're going to have movement of a solute against its concentration gradient. So again, we are now moving from lower concentrations to higher concentrations. Active transport helps to maintain the gradient between the cell and the interstitial fluid. So as we move, or as we look at passive process and we move down the concentration gradient, that actually decreases the gradient. With active transport, because we're moving it against the gradient and we're moving from low to high, we are actually maintaining that particular concentration gradient. And then the source of energy determines whether the movement is going to be primary or secondary. And we'll talk about that here in just a second. So with primary active transport, we're actually using the energy directly from the breakdown of ATP. We basically have a process known as phosphorylation, and that phosphorylation process takes a phosphate group and adds it to the transport protein. And as we look at the protein then itself, as it becomes phosphorylated, it actually goes through a shape change, and that shape change allows the movement of the ions or of the substance across the membrane. So let's look at the ion pumps that are involved here. Ion pumps are going to be cellular protein pumps that move ions across the membrane, and it helps to maintain the internal concentrations of the ions. So if we look here, again, we are utilizing energy. We're utilizing ATP, and you'll see ATP goes through the process and it gets broken down. So now we have the energy released from the bond that was formed with that phosphate group. But now we are left with ADP and this inorganic phosphate group. And now we'll see that calcium is able to come in and basically pass through this particular channel as it goes through a shape change, okay? So that is our active transport. We also look at the sodium-potassium pump, and this is kind of one of these examples. This is an exchange ion pump. And so what it does is it moves 3 sodium ions out of the cell and 2 potassium ions into the cell. So again, 3 sodium ions and 2 potassium ions. And both of these are going against their concentration gradients, because what we're going to find out, and we'll talk about this a little bit later, sodium has a concentration 10 times greater outside the cell than it does inside the cell. And the reverse is true for potassium. Potassium has a concentration 10 times greater inside the cell than it does outside the cell. And so as the sodium potassium pump works, it's moving these ions against their gradient. So it moves 3 sodium ions out of the cell, and it moves 2 potassium ions into the cell. And this allows the plasma membrane then to maintain or preserve the steep gradient for these particular 2 ions. Now, we are constantly moving these ions into and out of the cell. And we'll talk about the prevalence of, or the significance of these ions a little bit later as well. But the cell is continuously exporting sodium out of the cell and continuously moving potassium back into the cell. So if we take a look at the sodium potassium pump here, you basically see, and I know again this may be a little bit harder to see, but if we do look at it, we have sodium potassium pumps lining the entire length of virtually every cell of the human body. And so again, what we're looking at is, here we have the interstitial fluid. So this is the outside of the cell. Here we have the cytosol. This is the inside of the cell. Here's our sodium potassium pump. So here we require energy, so we require ATP. It's going to bind to the channel. It basically is broken down. So if we think about what is actually happening, this is a decomposition reaction. So we're breaking down ATP. and it becomes ADP, an inorganic phosphate. So as we break this inorganic, or as we break ATP down, we use the energy from those bonds. Now we have our 3 sodium ions that bind to it in this phase. The channel is now ready to pump. We go through the process of breaking down ATP. We now have ADP, and you'll see that the sodium ions are moving out of the cell. As we hit that point then, now we've opened up this little area, and this area allows our potassium ions to come in. So now potassium comes in and fills up its 2 spots. Again, the protein is now going to go back through a shape change back to its original conformation, where now we have the transport of the potassium ions back into the cell. And it's at this point then, once the potassium ions are released, the sodium potassium pump is now ready to go through the process again. Because again, potassium has been released, sodium can now come in, we break down ATP, we get ADP, an inorganic phosphate, the sodium is released into the cell, potassium binds to the channel, it goes back through a shape change back to its original conformation and is released back into the cell. And so you see that process, it's just a continuous process that is continuously happening throughout the cells as well as throughout the body. Now we also have secondary active transport. And secondary active transport actually moves the substances against the concentration gradient. However, the one thing that we're going to see is that secondary active transport doesn't directly utilize ATP. What it does is it uses the energy from the movement of a second substance down its concentration gradient, okay? So let's kind of talk about what that is. We're actually utilizing kinetic energy, providing the power to pump the other substance. And so what we see then is sodium is now moving down its concentration gradient. And so secondary active transport depends on the sodium potassium pumps for energy, okay? So it actually depends on the sodium and the potassium gradients that's provided by that sodium potassium pump. And we have 2 different types. We have symport transport and antiport transport. So if we're looking at secondary active transport and we look at symport, with the symport transport, we have 2 substances that are moving in the same direction, and these are called symporters. And therefore we have a symport secondary active transport. Now with antiport, we have the reverse. We have 2 substances that are actually moving in opposite directions. And these are called antiporters. And the process is called antiport secondary active transport. So if we look at these 2 processes here we have a symporter, here we have an antiporter, okay? You'll notice that we have our purple bubbles here. These represent our sodium ions, okay? Here we have glucose is represented in this case. And here we have hydrogen represented in the other case. With a symporter, the substances are moving in the same direction. Now remember, we're actually utilizing the concentration gradient of sodium and potassium that's been provided by the sodium potassium pump. In this particular case, we're utilizing the sodium gradient. So sodium has a higher concentration outside of the cell. With the sodium potassium pump, we're moving sodium from inside the cell to outside the cell. With secondary active transport, we're actually utilizing that gradient. We're allowing our sodium to flow through the cell or through the channel. And we have this symporter. This is where we utilize glucose. Glucose comes in and binds to the channel, and that allows glucose to flow into the cell because of the, because of the concentration gradient that's been provided for sodium. And so now sodium flows down the gradient, glucose moves into the cell, and then sodium is then pumped back out by the sodium potassium pumps. Now we also have the antiporter system. And this is where the ions are moving in opposite directions. So here again, we have sodium flowing into the cell, but now we have hydrogen actually moving out of the cell. So it binds to the channel and it gets moved out, okay? So again, this is secondary active transport. It doesn't directly utilize ATP, but what it does use is it uses the ATP from the sodium potassium pump that was present in primary active transport. And so therefore that gives it makes it fall into kind of that active category. So then we have vesicular transport. And with vesicular transport, this actually involves energy input to transport large substances across the plasma membrane by a vesicle. We typically are going to see membrane-bound sacs that are going to be filled with material. And these are organized into the process of exocytosis and endocytosis. With exocytosis, we have large substances that are secreted from the cell. So they're actually moving out of the cell. Therefore, the terminology exo, okay? We're moving substances out of the cell. It transports macromolecules that tend to be too large to be moved across the plasma membrane. And so what we find is that material then is packed within these intracellular transport vehicles or vesicles. And this vesicle and the plasma membrane then begin to fuse. And the fusion of the vesicle and the plasma membrane actually requires the ATP. From here, the contents are then released to the outside of the cell following the fusion of that vesicle to the plasma membrane. And an example of this is going to be the release of the neurotransmitters from our nerve cells, okay? And so we'll talk about that when we get into later chapters dealing with muscle contraction and action potentials. So here's our process of exocytosis. You'll see here we have a vesicle that's got a bunch of substances in it. Again, these are typically just going to be too large to directly pass through the plasma membrane. And so you'll see that the vesicle nears the membrane. Here the vesicle fuses with the plasma membrane. So it's starting to join the plasma membrane itself. As it becomes a part of the plasma membrane, it releases the contents out of the cell and they are able to exit. If you take a look at the vesicle, you'll notice the vesicle has kind of a similar structure, this phospholipid bilayer, so to speak, that's very similar to the plasma membrane. And so as that vesicle basically joins or merges or fuses with the plasma membrane, it actually becomes a part of the membrane until it becomes pinched off and gets pulled back into the cell. Endocytosis is where we actually utilize cellular uptake of large substances from the external environment. So here we're actually taking things into the cell. The steps of endocytosis are similar to exocytosis, but they actually occur in reverse. And so a few reasons we would have endocytosis would be for the uptake of materials for digestion, so things that the body or the cells need to break down. The retrieval of membrane regions from exocytosis, so things that need to get pulled back into the cell. And the regulation of the membrane protein composition that alters or that needs to be altered for cellular processes. So there are three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. With phagocytosis, we can consider this cellular eating. And so what happens is that the cell actually engulfs a large particle from the external side of the system, and it pulls it into the cell. What we see is that the cell will actually create large extensions called pseudopodia. And so they're basically considered false feet. That's what pseudopodia means, is false feet. And so they basically extend out, they surround the particle, and then they enclose it in a membrane sac. The sac is then pulled into the cell, it's internalized, and the contents are digested after that new vesicle fuses with a lysosome. I know we haven't talked a lot about different parts of the cell yet, But one of the components of a cell is a lysosome, and these lysosomes contain enzymes that help to digest certain substances. Now, only a handful of different types of cells will perform this type of process, but one of the major ones is going to be a white blood cell. And so a white blood cell can go in, it's going to engulf some sort of invader, typically a microbe of some sort. It pulls it in and it's able to digest it and break it down with the use of a lysosome. So here we have our cell on the inside. Here we have the pseudopodia. Again, I said these are basically extensions of the cell, and they kind of wrap around. And so these are the false feet as they extend around some sort of particle or invader or a microbe. As it then pulls it in, you now see that we have this newly formed vesicle. And with phagocytosis, we're now taking this particle and we're actually going into and digesting it. And again, we'll see some sort of lysosome that would come over here that would have a whole bunch of digestive enzymes, and it would actually fuse with this particle or this vesicle in order to digest the particle that's inside it. We also then have penocytosis. Now, penocytosis is cellular drinking. And with penocytosis, we have this internalization of droplets of interstitial fluid that contain dissolved solutes. In this case, we see multiple small vesicles are formed, and it is performed by most cells. So if we look at penocytosis, again, somewhat similar to phagocytosis, we don't quite see the full extensions, the false feet that extend out, those pseudopodia. But what we do see are these invaginations of the plasma membrane. And again, all these different colored specks out here are different substances within the interstitial fluid that the body is actually taking in. So oftentimes these can be just plain ions or just fluid that the body really needs to take in. And so you'll see the invagination. You'll see as it pinches off, we now have the new vesicle, and that vesicle gets pulled into the cell. Our last one is receptor-mediated endocytosis. And receptor-mediated endocytosis uses the receptors on the plasma membrane to bind molecules within the interstitial fluid and bring the molecules into the cell. What it does is it enables the cell to obtain bulk quantities of a substance. And so it can help transport of cholesterol from the blood to the cell. And cholesterol, we know, is kind of a bound low-density lipoprotein, or is bound to those low-density lipoproteins. And it moves from the blood to the interstitial fluid and then binds to the LDL receptors in the plasma membrane. Our LDLs then are internalized into the cell. So here's our example of receptor-mediated endocytosis. Here you'll see that we have this clathrin-coated pit, and it forms a clathrin-coated vesicle. Again, you'll see that here we have our ligand receptor complex in which we have some substance come in, a specific substance onto the receptor, so only certain things are going to be able to get in. And so because of that, these substances come in and bind to those particular receptors. And this is how we pull some of these substances in then. Again, receptor-mediated endocytosis is very specific. And so it does the same thing as what the other processes do. It pinches off the plasma membrane and it pulls that substance into the cell. But the biggest component here, the biggest factor, is that we have these ligand-gated, or I should say ligand receptor complexes that are specific for certain substances. So here we have now where we look at the resting membrane potential. So this is kind of a transition from movement of substances to now starting to look at the electrical components of the cell. Now the plasma membrane establishes and maintains an electrical chemical gradient, which allows us to cause or form a resting membrane potential. And this resting membrane potential is essential for muscle and nerve cell function. So let's look at our resting membrane potential. So the resting membrane potential really is the electrical charge difference at the plasma membrane. The membrane potential is the potential energy of the charge difference. And so more specifically, as far as the resting membrane potential goes, it's the potential when a cell is at rest, okay? And so the membrane potential then can actually change as the cell becomes active. For example, with a nerve cell, as it sends a action potential or a communicating impulse from one cell to the next. There are two conditions of the resting membrane potential. We have an unequal distribution of ions and molecules across the plasma membrane. We have more potassium in the cytoplasm than in the interstitial fluid, and this is about 10 times greater inside the cell. You'll notice this is bolded. You will want to know this, not necessarily just because it may be on an exam, but you're going to want to know this because the better you understand this statement and this statement, more sodium in the interstitial fluid than in the cytoplasm, and it's 10 times greater outside the cell. The better you understand these two statements, the better you're going to have an understanding of muscle contraction and the action potential of the nervous system. So please make sure that you commit these two statements to memory. Now, this unequal distribution, obviously we know, is due to the sodium-potassium pumps as it continues to pump potassium out of the cell, or excuse me, sodium out of the cell and potassium back into the cell. And then the other condition of the RMP is we have unequal relative amounts of positive and negative charges. These are going to include different negatively charged protein anions, as well as our negatively charged chloride ions that we have inside and out of the cell. And so we have more positive on the outside than what we do on the inside of the cell. So if we take a look at our resting membrane potential then, we're basically taking a voltmeter, putting the ground outside the cell, and really taking a measure of the voltage on the inside of the cell. You'll notice that the voltmeter here reads -70. This is kind of the standard resting membrane potential for the majority of cells, particularly the couple cells that we're really going to talk about with muscle cells and nerve cells. But you'll see down here that that resting membrane potential can range from about -50 to about 100 millivolt, -100 millivolts, depending on the cell itself, okay? But again, you'll notice that we represent a lot of the different types of channels here. We have leakage channels for sodium and potassium. We have our sodium-potassium pumps. Inside the cell, we have negatively charged anions that contribute to a more negative inside part of the cell. And so again, just remember that we're taking that measure from the inside of the cell. So the inside of the cell is more negative than the outside. So when we try to establish and maintain our RMP, by now we should know that the most important ions involved are sodium and potassium. And sodium-potassium movement really depends on the electrical chemical gradient. So let's take a look at the role of potassium first. The role of potassium is the most important determinant in the specific value of the RMP. We're going to find that potassium moves down a steep concentration gradient through leakage channels from the cytosol to the interstitial fluid. Okay, so because we're moving through leakage channels, we're moving down our concentration gradient, okay? And so it's actually moving out of the cell. We have negatively charged proteins that remain inside the cell. And so the electrical chemical gradient has a positive charge outside, and that actually repels the movement of the potassium ions out, okay? The negative charge on the inside of the cell actually begins to attract back some of those potassium ions that we're trying to move out. And so what ends up happening is potassium will continue to move in and out of the cell until equilibrium is reached. And once equilibrium is reached, that's actually where we have an equal movement in and out of the cell of just regular potassium ions. Okay. So again, this kind of electrical chemical gradient is developed because of potassium. Now, what's the role of sodium in the RMP? Well, sodium diffuses into the cells from the interstitial fluid to the cytosol simultaneous to the loss of potassium. So again, both of them have a positive charge. So to a certain degree, the electrical component is balanced out. It's going to enter through our sodium leakage channels, and again, it's moving down the concentration gradient as we move through the leakage channels, because leakage channels are always open. And then it's pulled by the electrical gradient. Okay. Now, our leakage channels prevent as much potassium into the neuron as potassium out. Okay. And then this then leads to the inside becoming a little bit more positive. Okay. So now we're starting to make a little bit of a shift with that resting membrane potential. So as we start to have these slight changes in charge, and we have leakage channels allowing sodium to come back in, which makes it more positive, how do we maintain the electrical chemical gradient of the RMP? Well, that's where our sodium potassium pumps come into play. It's going to help to maintain the potassium and the sodium gradients following their diffusion through those given leakage channels. We therefore then pump sodium out and we pump potassium back in. Okay. Now I'm going to scroll back one side. Again, we notice the inside becomes a little bit more positive due to the role of potassium in this case. And so what ends up happening? We actually are pumping 3 sodium out to only bringing 2 potassium in. And so we're actually forcing the inside of that cell to become a little bit more negative because of that. Again, sodium and potassium are moved in opposite directions, and they are both moved against their concentration gradients. So let me scroll back here. So that's kind of the overall idea of what the resting membrane potential is. Again, you really have to look at the movement of potassium and sodium as they move in and out of the cell. And again, coming back to the sodium potassium pump as being one of our significant components of helping to really maintain those concentration gradients so that sodium stays have a higher concentration outside the cell, and so that potassium maintains a larger concentration inside the cell. So then let's look at some contact between cells. So direct contact between cells is really important for some of our cells to function, specifically some of the cells of our immune system. It's going to distinguish our normal cells from unwanted cells. We also have to look at the sperm and the oocyte. So we basically have the egg with a unique glycocalyx that's identifying it as an egg cell. And this therefore allows the sperm to recognize that particular egg or those egg type cells during fertilization. We also need to be able to identify cells for cellular regrowth following injury. In this case, we're going to find that damaged tissue is replaced by cell division in the epidermis. So damaged tissue is cleaned out. We also have cellular contact as cells are identified to help prevent overgrowth, so that once cells basically fill in the area that they're supposed to, they don't continue to grow. So again, that's why we have this importance of direct contact between cellular tissues. So that leads us to ligand receptor signaling. And so most of our cell communication occurs through ligands, which again, ligands are going to be molecules that bind with macromolecules. Neurotransmitters are also a form of a ligand, as are hormones. So neurotransmitters from nerve cells and hormones from our endocrine cells. And we also see that they're important for controlling growth, reproduction, and other cellular processes. So we have three different types of receptors that bind ligands. We have channel link receptors, enzymatic receptors, and G protein coupled receptors, and we'll touch on each of those. So channel link receptors permit the ion passage into or out of the cells, okay? And so you'll notice over here that we have this channel link receptor. And so we have these ligands that come in and bind to the channel. Note that the channel is very specific to a specific ligand. As the ligand binds to it, it basically causes a shape change, and that causes the channel to open up, allowing these ions to flow through the channel. In this case, it occurs in response to the neurotransmitter binding it, and this is going to help initiate electrical changes to the RMP in muscle and nerve cells. So we'll talk a lot about channel link receptors and ligand-gated receptors as we get into muscle contraction and action potentials. Our next one is an enzymatic receptor. Now, enzymatic receptors, in this case, we have kind of a protein kinase enzyme. So you'll notice that we have the protein here, or the enzyme here. You'll notice that here comes the ligand. It's going to bind to this particular protein kinase enzyme here, this particular protein. And you'll notice that here we kind of have this enzyme actually change shape. So it's activated to phosphorylate other enzymes within the cell. And it provides this mechanism for altering enzymatic activity. So again, note we have the ligand come in, binds to the particular channel. You'll notice this inactive protein kinase enzyme at one side of it. Once the ligand binds to it, you'll notice that now we have an active protein kinase enzyme, and it's now actually able to phosphorylate other enzymes and actually allow other enzymes to be turned on or off, okay? So again, it's just a process of activating additional processes. Our next one is a G protein coupled receptor. And what a G protein coupled receptor does is it indirectly activates the protein kinase enzyme. So let me take a scroll back once. So in this case, we directly activate the kinase enzyme on the particular protein. With a G protein coupled receptor, we kind of do this indirectly. So here you'll see we have the protein, we have the ligand come in, it binds to the receptor, it causes this shape change. And what this does then is it binds to and activates this particular G protein, okay? And you'll see down here, GTP binds to the G protein, causing G protein activation. So now this activated G protein moves over, and now it can actually cause a opening of a channel, or it can come down here and it can cause an effector protein to become activated, and it can stimulate a second messenger. That second messenger is going to come over here, and it's going to activate this particular kinase enzyme, which now can do the same thing that our previous receptor just did in the previous slide, and now it can turn on or off a different enzyme. So basically, we're looking at very similar process with the enzymatic receptors and the G protein coupled receptors, in the fact that these two particular messengers or receptors essentially have an end goal of trying to turn on or turn off a particular enzyme, okay? And so let's just use G protein as our code word, attendance code word for today. So again, G protein will be our attendance code word, and this will be the end of this section of chapter 4.